genetics document that I gave you. We'll take a few notes on day 2 and go over who Mendel is and what complete dominance means. And then there's some homework that we'll have some time at the end of class for you to get started that will be due tomorrow. So you'll see our essential question for the day. We're trying to answer this question: what is a genotype and what is a phenotype? And what are dominant and recessive alleles? I guess that's actually like 4 questions instead of 1, but I tried to write it as 1. And hopefully by the end of today, or at least by the end of the time you do your homework, you'll be able to say, I can name a genotype based on what alleles an individual has and tell what the individual will look like. It's phenotype. So we've got a lot of vocab words that we're going to go over today. And I'm hoping that some of this is review for you if you remember genetics from 7th grade. If not, well, I'm happy to teach you all of it all over again. So I'm going to stop sharing here. Before we get into the notes, I want to explain what is behind me. Because if you were here in school with me, this is what you would see on my board at the beginning of class. I would go over these vocab words and why I drew this like this. So I want to start off today by doing my best to explain what it is that we're looking at, some of these vocab words today. So I have my handy dandy star pointer here. So we're going to start up here at the very top with this vocab word, alleles. Can everyone say alleles? I know you're in your room saying it quietly, but let's all say alleles.
Alleles.
Alleles. So there are 2 different types of alleles out there. There are dominant alleles and there are recessive alleles. Okay, and we use letters to represent alleles. And what they are, they're different versions of a trait. So in my example I've written on the board, it's flower color. That's my trait. So you can either have a dominant allele for flower color, which represents the red color, or you can have a recessive allele for flower color, which represents the blue version of the flower. Okay, so our dominant allele is red and our recessive allele is blue. Now, everybody has 2 alleles for every trait. I know you guys don't have the gene for flower color because you're not flowers, but everybody has 2, one from their mom and one from their dad. So the next vocab word is genotype. So a genotype is what alleles do you have? Which ones did you get from your parents? So there's 3 different types of genotypes. You can be homozygous dominant and have 2 dominant alleles. So this prefix right here, homo, means same. So whenever you see the prefix homo in front of a word, it means same. So when we're talking about having 2 of the same alleles, homozygous dominant, or you can be homozygous recessive and have 2 of the small recessive alleles. The 3rd type of genotype that there is heterozygous. So hetero means different. So in the heterozygous individual, they have 1 dominant allele and 1 recessive, different alleles. Okay, they have 1 of each. Hetero means different. So when you say the word heterozygous, that means 2 different alleles. When we say the word homozygous, we must then say dominant or recessive, because there's 2 types of homozygous. Okay, and I'll get into that a little bit more in just a bit. So your genotype determines your phenotype. This is our last vocab word up here. So a phenotype is what you look like, and it's based on your genotype. So today we're only talking about complete dominance, meaning that one allele completely dominates over another. So if our genotype is big R, then our phenotype is going to be red. Okay, see how this big R leads to the red flower. If our genotype is little r, our phenotype is going to be blue, the recessive trait. And if our genotype is heterozygous, big R, little r, then our phenotype is going to be red. And this red color is because anytime the dominant allele is present, it's going to show up in the phenotype. Okay, that's what it means to be dominant. The dominant allele covers up. It masks the little r so that only the dominant color shows up. Okay, so now that I've explained that, I'm going to go over it a little bit slower, and we're going to go through it on your notes. And I just realized I wanted to do the warm up with you first, but we'll keep this all in your mind right now. And then we're going to warm up from what we did yesterday, and then we'll get back to this. I apologize for my timing, especially since Mr. Austin's recording this. All right, so let's open up your notes. So today is genetics day 2. And I really did mean to go over this warm up before I went over the vocab words behind me, but maybe it'll help for you to have them lingering in your mind right now as you go through this. So yesterday we went over some vocab words that you're going to see in this warm up. Gamete, these words, haploid and diploid. So what is a gamete? So if I asked you, let's say I was giving you a quiz right now, and I was asking you, what is a gamete? What would you answer? So at this point, if you wanted to, you could scroll up to your notes from yesterday and go, I took some vocab notes on gametes. Ms. Klepper talked about them up here. You could kind of look around and try to find the answers. So yes, it is a sex cell. Some people are direct messaging me that it is a sex cell. So it's either a sperm if you are a male, or it is an egg if you are a female. A gamete is a word we use to describe a sex cell, sperm or egg. What word describes the chromosome number that you find inside of a gamete?
Is it haploid?
It sure is. Haploid. So remember that egg and sperm have half the number of chromosomes as you can that you'll find in a body cell, which I might have just given you the answer to number 3. What is a somatic cell?
Cell that's found anywhere else in the body?
Yep. So it's we call them body cells. Like soma means body. And what I mean by this is any cell in your body besides sperm and egg. So it could be a liver cell. I'm just going to give you some examples. Liver cell, muscle cell, brain cell. We could keep listing cells like all day. There's so many different types of cells. But a somatic cell is just any other cell in your body that's not an egg if you're a female or a sperm if you're a male. And then what word do we use to describe the number of chromosomes in our somatic cells?
Diploid.
Diploid, yep. All right. Who remembers the magic human diploid number?
46.
46, yep. Faith, that's a little few too many, 96. It's 46. So we have 23 pairs, one from mom, one from dad, for a total of 46. So our haploid number is
23.
23. So we find 23 chromosomes in sperm, 23 chromosomes in eggs. And then how many chromosomes did you get from your mom then?
23.
Sure did. And from dad?
23.
23.
Yeah. In a perfect world, everybody gets 23 chromosomes from mom and 23 from dad. And I just want to remind you of what these pictures are. I showed this to you yesterday in the vocab slides, but I gave you this here because I wanted to show it to you one more time. So what this is a picture of here is a picture of somebody's chromosomes. So a picture was taken and then the chromosomes don't line up this nicely. People have to actually say, they have to like find chromosome number 1 and put it next to 1 and 2 and find it. And they're able to do this by looking at these little lines. See, if I zoom in on here, there's little lines and they can tell which one is which. So this is somebody's job, is to take a picture of chromosomes and put them into this perfectly organized little picture. And women who are pregnant get these pictures taken sometimes of their baby to make sure that it has the correct number of chromosomes, because they can see there should be 1 chromosome from mom and 1 from dad for all 23 pairs. So pair number 23 is your sex chromosomes. This is what determines your sex. And we talked about yesterday, this XY would be a boy. Females have 2 X's, so this Y would be another X if it was a female. And this is what it looks like, that your chromosomes look like in your diploid somatic cells. Now, in a sperm or egg, you only give your offspring just 1 of your chromosomes. You don't give them both. You give them just 1. So when meiosis is creating gametes, haploid cells, you only give your offspring 1 of each of your 23 pairs. All right. So today we have some vocab words, and I'm going to go over these again at the end of class for those of you who want to see my vocab slides, but I'm probably going to run out of time for some of you who will want to just start working on your homework. And I just want to remind you that these notes, I'm updating them every day and you have the link to them. So if you want these notes later, like you don't want to get them today, you can do that. Also, the vocab slides are available on Canvas as well. All right, so now getting back to what I started talking about with my beautiful picture behind me here that I've drawn. We're going to talk about these terms now and take some notes on them. But to get us started, I want to show you a video and briefly mention who Gregor Mendel is. So Gregor Mendel, we call him the father of genetics. And the reason we call him that is because he is the person who discovered the idea of dominant and recessive alleles. He was the one who noticed that one version of a trait can dominate and cover up another. He was the one who realized how you get a recessive trait to come back, reappear in the offspring. And so even though he wasn't really working in a lab, he was working in a garden. He was a monk he actually ended up being a monk, who was working in a garden and doing experiments on pea plants. And he kept documents of all of his pea plants, and this is how he discovered the stuff that we're going to be talking about today and tomorrow. And unfortunately for Mendel, the scientific community wasn't very nice to him, and they didn't really give him credit for what he did until after he died. So he discovered all of these rules that we're going to go over here, and the rule, kind of our ideas of how genetics work. Even though he didn't really get recognized for it until after he was gone. So I'm going to show us this quick video here. There's a link if you want to watch it again. But I think it's worth watching, and it goes over these vocab words that I've already gone through with you in just another way. So I'm going to pull this up. I'm going to ask if you have your video on, if you can just turn it off for right now so that my computer can process this video for you. And here we go.
Between 1856 and 1863, Gregor Mendel, an Austrian monk, grew and studied peas. That probably doesn't sound that exciting, but what he learned about genetics changed everything. Let's look at what he did. Mendel bred pea plants, looking for many different traits like color, or wrinkly or round seeds, or tall or short plants, among other things. Once he had a purebred trait, he would run an experiment. Mendel crossed two purebred traits. We'll look at green versus yellow-colored peas. When the offspring grew, he found that all of the peas were yellow. He couldn't understand why the green trait disappeared. Why didn't the peas have a 50-50 chance of being green or yellow? To explore this further, he took two peas from the first filial, or F1 generation that was just made, and he bred them together. Yes, this is inbreeding, but they're peas, so no one seems to mind. When the second filial generation arrived, F2, 75% of the peas were yellow, but 25% were green. The green peas seem to disappear in the F1, but magically reappear in the F2 generation. What could be happening? Mendel decided that there were factors, which we now call genes. The genes could be either dominant or recessive, and dominant alleles or traits mask recessive alleles. In this case, the yellow is dominant and the green is recessive. So the purebred parents made offspring that had green and yellow alleles, but only the yellow showed because it was dominant. And when the F1 generation bred, there was a chance that they could both give the recessive allele to the offspring, so the green peas showed up again. If you could look at the chromosomes in the pea plants, you would find the gene for seed color. A gene is a section of DNA that controls a trait. The different seed colors have different alleles, which are variations of a gene, like green or yellow. Allele is a fun word to say, and it's actually even more fun to write in cursive, but you can try that next time you're bored. Now you get two alleles for every gene, because you have a set of chromosomes from mom and a set of chromosomes from dad, and the same is true for plants. When you write alleles, dominant alleles get capitalized letters, and recessive alleles are lowercase. If a dominant and recessive allele are together, it's called heterozygous. Hetero means different, and the alleles are different, so that makes sense. If the alleles are the same and dominant, we call that homozygous dominant. Homo means same. If the alleles are the same and recessive, we call them homozygous recessive. The letters we choose here are arbitrary, but it's good to use letters that are very different when you view capital and lowercase versions, otherwise it can be confusing. So letters like O aren't good, but letters like Y, T, or R are great. In pea pods, green is dominant over yellow. If these are the genotypes or allele combinations, what are the phenotypes? Having just one dominant allele of the gene will make it green in this case. So there are two green pods and one yellow pod. Now this is pretty simple, but let's try thinking about this in terms of making gametes. Assuming tallness is dominant, what would the genotype look like for a man who's homozygous for tall? And what are the possible gamete combinations? Now, because tall is dominant, this would be a homozygous dominant genotype, 2 capital letters. Again, you can choose any 2 letters you like, but I'm going to use T for tall. When gametes form, only half of the DNA is in each cell, so only one allele can go to each of the sperm that are formed. There's only one possible gamete combination in this case, capital T, the tallness gene. But let's try another case. What would the genotype look like for a woman who's heterozygous for tall? And what are the possible gamete combinations? Now, heterozygous means different alleles, so this will be written with a capital and a lowercase t. This also means that there are 2 possible gamete combinations, an egg with a big T for the tallness gene, or an egg with a little t for a short gene. Heterozygous traits will have more variation in their offspring. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter at SciencePet.
All right. So I'm going to go back to your notes here. So again, if you want to watch that video again, because it always helps to watch something more than once, the link is in today's notes. So please feel free to go ahead and watch that video again. So we're going to go through some of these ideas that were talked about in the video and that I went over on my board here. So first of all, the first rule of genetics is that genes come in pairs. So you have 2 alleles for every trait. That's how you get a trait. That's how you inherit a trait. You get one of those alleles from your mom and one from your dad. All right, rule number 2 says that one allele in a gene pair may mask or cover up the expression of the other allele. And that allele that shows up is called the dominant allele. Gosh, the dominant allele. The one that's being hidden is then called the recessive. And when Mendel discovered this idea of dominant and recessive, he said, "I shall call this the principle of dominance." And that's what he called it. And that's what we still call it, the principle of dominance, is that one version of the trait dominates over the other and hides it when present. All right, rule number 3 is that when sex cells form, so sperm or eggs when they're made, during the process of meiosis, each sex cell that forms gets one allele for each gene, only one. And Mendel said, "I shall call this the principle of segregation." Pretty sure he spoke English. Maybe he didn't. He was Austrian. He probably spoke Austrian. German? Austrian.
They speak German.
So, ich shall, I don't know. I don't even know
Just
the word.
sound aggressive. Just whatever you say it in
I
a
shall call this
throaty, angry
principle.
voice.
So what the principle of segregation means is what she was showing in that video, is that if you are a heterozygous individual, this is your diploid somatic cell. Let's say this is your somatic cell. When you form sperm or eggs in meiosis, you only give one letter to your offspring, either the big T in this case or the little t. These are my sperm. Segregation means that the pair of alleles that you have are going to separate during meiosis. This will be really important tomorrow when we start looking at Punnett squares. All right, so when we write genotypes in genetics, we always use capital letters to represent dominant alleles. And we use lowercase letters to represent recessive alleles. I always seem to capitalize the word dominant and I never capitalize the word recessive. It's just, I don't know, it just happens. Thank you, Ethan. Ich werde dies das Prinzip der Segregation nennen. That was, don't tell my German teacher that I just pronounced it like that.
Jawohl.
Jawohl. Nein, das ist nicht gut. All right. Now, if an individual has two of the same alleles, so we use the prefix homo. Homo means same. We call that individual homozygous. So you can either be homozygous dominant and have two capital. So this would be homozygous dominant. Or you can be homozygous recessive. You can't just say homozygous, is what I'm trying to get at. You have to say recessive or dominant after the word homozygous. Okay, because otherwise the person's going to go, well, which homozygous is it? Because it can be either dominant or homozygous recessive. So you have to, after you use the word homozygous, you must then say dominant or recessive. All right. Next up is if an individual has two different alleles, and the prefix for different is hetero, then we call that individual's genotype heterozygous. So big R, little r. If you're calling somebody's genotype heterozygous, you don't say anything else after that. Okay, heterozygous means two different alleles. So you don't use any other words after that word. But when you say homozygous, you must then say dominant or recessive, because otherwise we have no idea what you're talking about. Okay, heterozygous already means one thing, that you have two different alleles. All right. Next up is phenotype. So this vocab word means how an individual appears, what they actually look like. And it depends on their genotype. Okay, so a phenotype depends on a genotype. All right, so here's where you get to practice and see if you so far are understanding what I've been talking about. So for my example here, we're using the letter B to represent hair color. Okay, so hair color is a trait. We're using B to represent that trait. If we're using the uppercase B to represent the dominant hair color of brown, and lowercase b represents hair color of blonde, then what are the genotypes for these three individuals based on their genotypes? So what color hair would they have? Some people are direct chatting me. Yep, so big B would represent brown hair, right? Because there's two dominant alleles, two dominant brown alleles. And big B, little b would also be brown hair. Because remember, anytime you have the dominant allele, the uppercase allele, it's going to show the dominant trait. It covers up this little b. It's like the little b is not even there. Okay. So then the last one, little b, is homozygous recessive. And this one would show up as blonde hair. To be perfectly honest, hair color actually is controlled by more than one gene. That's why we have all different colors of hair, not just brown or blonde. So this is just a simplified example. Don't get too caught up in that. All right. So I want to show you what your homework looks like and what you're going to be doing for your homework. Your homework is page 8 and 9. So the next 2 pages on this document. So the first part, you're just reviewing the vocab. So you can look in your notes and answer those questions. And then down here, you're going to start using some of this vocabulary that we just went through. So you have a chart here. And what you're going to do first is you're going to look at the alleles, the genotype that this individual has. And you're going to tell me, what word do we use to describe that genotype? So is big T, little t, is it heterozygous, homozygous dominant, or homozygous recessive? So you're going to write that down right here. Then you're going to use this key up here to determine what the phenotype is. Okay, so there's different letters that represent different things. Like T is representing hair texture, whether you have curly hair or straight hair. R is representing eye color. Y is representing leaf color. And B is representing eye color, again, for 2 different colors. So as you're going through this, you're going to have to reference this key up here to determine the phenotype of each of the individuals. Somebody's asking me, can I abbreviate and use homo-het? I would really like it if you wrote this out. And the reason I want you to do that is because every time you write it out, it's helping you remember it. And I'm just gonna also point out the scissors tool. Why does it keep happening? You have a scissors tool up here too. So not to give you like an out or anything, but it's reinforcing to write it out. So it's only a couple things you need to write it. It's gonna be okay. So you're gonna do that first. And then on the second page, oops, I didn't mean to leave this out here for you. You're looking at a trait for ear type. So earlobe type. So there's a type of earlobe that's dominant over another type. And if you can see me right now, I don't know if I can pin myself. Oh, I'm having a hard time pinning myself. Here. So if you guys can see my ears right now, I have a free earlobe, meaning that my earlobe is floppy, like I can flop it back and forth. An attached earlobe is when your earlobe actually is attached to the side of your head and you don't have this floppy earlobe like I do. And so having a free earlobe like mine is dominant over having an attached earlobe. Okay, so using the letter E, you're going to answer the following questions. So I'm going to just point out that there are quite a few vocab words here that you're going to need to make sure you understand in order to answer these questions correctly. So it's saying if the father is homozygous recessive for the earlobe trait, what is his genotype? So you're going to have to make sure that you understand those. And then it's going to ask what is his phenotype based on that. Number 3 and number 6 are getting at what alleles can each of these parents pass on to their children. So for that, I'm going to point out that you have this picture here, and I talked about the principle of segregation. So whatever the father's genotype is, that's going to determine what possible alleles he can pass on to his offspring. Okay, and the same thing goes with mom down here. If mom is heterozygous, what alleles can she pass on to her children? I'm just gonna, again, remind you of this image right here. There is a bonus question down here, number 7. That one you might not be able to answer. I'd love you to try. I want everyone to try it. If you get it wrong, we're not gonna mark you down for it. The question is asking if it's possible for these two to have a child that's homozygous dominant. And tomorrow we're going to talk about Punnett squares. So tomorrow you should be able to answer this question, but for today, I just want to see if anyone can figure it out. Okay. You should be able to answer number 8 and 9 just fine, but 7 might be a little bit of a challenge. Okay. So that's all that I have for you, except for if you want right now. So there, you have a couple options